thank you so much for coming, and thank you for being here. Thank you so much for this invitation. This is exciting. So I've been at RIT now for 10 years, and I took my first deaf student, research student, my first year at RIT. And I had only had very limited experience working with deaf and hard of hearing students in my classroom. And I said, oh, sure, you know, I'll have a deaf student do research in my lab. And because I was really confident in my mentoring skills. I felt like I'm a really good mentor. I was a mentor as a grad student. I mentored students at my postdoc. And so Todd Pagano asked me to take a co-op. So I said, sure. So I took that student. It was a disaster. It was a disaster. I found out that the student was scared of me, which surprised me because I don't think of myself as a scary mentor at all. So I felt really bad. It just made me feel awful. I said, okay, I kind of went at it as I'm gonna just treat all my students the same, like I can do this, everything will be fine. But it wasn't fine, and it made me realize I still have so much to learn. I mean, I was only a first year anyway, but I have so much to learn. And so I said, I'm gonna take more deaf students so that I learn how to be a better mentor, specifically to deaf students, because we have such a large deaf population at RIT. Why would I not want to be a better mentor, specifically with deaf and hard of hearing students? And so it became, and it's been so much fun to get to know all of my research students, but I've had amazing deaf students like Kara, who's right there. And Kara has been one of my, has been with me one of my longest students. I started working with Kara her freshman year, first semester. She came up to me. She joined the group really early on. So for the whole 4 years she was in college, Kara worked for me as a research student, and now she's getting her master's with me. So I couldn't be luckier to have her for so long. So I started off at Colgate University, and I got my bachelor's degree in physics and math. Go Colgate. I loved it at Colgate. I loved being at Colgate. It's a beautiful campus. It's not too far from here. I was definitely more a physics person than I was a math person. The math department was pretty big, but for physics it was so small that I felt like I belonged with the physics department, and I got to do a little tiny bit of research, but overall I had just an amazing experience. But I knew pretty quickly that I didn't necessarily want to continue on in straight physics, and so I had a little bit of a path to take. I put this in here because at RIT, it's very common for students to get research experience, right? You guys both had research experience, multiple different research experiences over the summer. So what did I do during my summers? I was a waitress at Friendly's, and I taught dance classes. So I did very little in professional development. I really didn't, because I just didn't think, I'm like, I want to make some money so I can have spending money in college. I knew I was studying abroad, so I wanted to make money for my study abroad. But I will tell you that I did learn a lot from being both a waitress and a dance teacher. As a waitress, you really learn to multitask and work with people, right? Because you're working with people. If you want them to give you a good tip, you got to be a happy, friendly waitress. And I really learned to multitask because you're doing 1,000 things at the same time. As a dance teacher, I learned how to teach. It was my first real experience teaching, and so I learned, even though it was dance class and it was to mostly little kids, it really helped me develop kind of my style of teaching, which I think was really important because you don't get a lot of teaching experiences before being a professor. So that was actually really nice. Like I said, junior year of college, I studied here, Cardiff University. It's in Wales and in the UK, and at Colgate, 75% of students study abroad. So most students do study abroad. So it's a cultural thing at Colgate, and so I studied abroad. I went with the science department. Most of the other students were neuroscientists. But it was an amazing learning experience, being immersed in a different culture. Not just going on vacation and spending a week or two there, but really like taking classes, we lived with, in the dorms, and just being immersed in a different culture was it was just such a great learning experience for me, and I loved it. It was probably the closest I'll ever get to being in an environment where it's hard for me to understand what's happening around me, because Welsh people have the thickest accents. I could not understand what my teachers were saying. I feel like I needed an interpreter from Welsh. They spoke in English, but it was just such a thick accent, and I was like, it's hard for me to understand. But we got through it, and I got to travel a lot. The one really important thing that I got out of my study abroad is that this was second semester junior year. I still had no idea what I wanted to do, none. I kind of thought, okay, I might as well just keep going to school or do something, because I can postpone real life as long as I can. So I did that. And I talked with my advisor. So one professor brought us to Wales, to Cardiff, and he said, okay, so you're a physics major, but you don't really want to do physics. Why don't you think about biophysics? I had no idea what biophysics. I'm like, oh, okay, biophysics, that sounds interesting. I'd never even heard the term. I had no idea what it was. And at that moment, after that conversation, I applied to all biophysics PhD programs. I did not even bother, don't ask me why, I didn't even bother to really look up what biophysics really was. I asked a couple people, but I felt really silly asking, because I'm like, I should know what this is. But I did, I asked a couple people. But the problem with biophysics is it's kind of nondescript. It's hard to define biophysics because it's so many different things. So really what it is it's a combination of physics, biology, and chemistry. It's kind of this smushed together thing, right? And so really what I did, what was the beauty of it, is I was able to use all the training that I had in physics to solve biological problems. And then I learned a lot of the chemistry kind of along the way. So, it was really, to me, it was almost like a triple minor doing biophysics, is you got to learn a little bit of chemistry, a little bit of biology, and a little bit of physics, and kind of pushing that, figuring out how you were able to use all those things to solve problems. Thankfully, even though I had no idea, I got into a couple biophysics programs. I applied to straight physics and some biophysics. And so I got into, and I only interviewed at the biophysics places. And it was just wonderful. And I really lucked out that I ended up loving biophysics, but it could have ended up really not going well. That the summer in between my junior and senior year, I decided, okay, I'm going to apply to these biophysics programs. I should probably get some lab experience. That was recommended to me. I asked my aunt, who worked at the University of Rochester, to get me a position, just like an internship. And so in addition to waitressing, I also spent a few hours a day in this lab. And so my first research experience was patch clamping cells, which is basically taking a glass pipette, and sticking it into a cell, and then putting a voltage across the membrane of the cell. And what did I do that summer? I basically sat on a stool and watched someone patch clamp cells. That's what I did the entire summer, is sit on the stool. That was and I watched this person patch clamp. That's all I did all summer. It wasn't not the best experience. It was definitely not hands-on. So not the best experience, but at least it kind of got my foot in the door a little bit. It got me a little bit of something. So I went on in biophysics. I went to University of Rochester right out of college, and I took biophysics classes. I learned what biophysics was. I was pleasantly surprised that it was kind of a perfect fit for me because of my physics background. Most people go into biophysics from biochemistry or from biology, and very few go in my program went from physics. So I was, I think, one of the only physics majors who went from physics to biophysics. So I did have to catch up a little bit on the biology and the chemistry. But I did so, and I really enjoyed it. And I really loved it. So in my program, we did 3 lab rotations. The great thing about it was you got to try out different labs before you had to commit to who you're going to work with for the rest of your PhD. So I usually recommend applying for programs that have these lab rotations. Some of them don't, like some of the straight chemistry programs don't have lab rotations. You just kind of pick a lab without even working in their lab first. That's really tough. I was able to work in these 3 people's labs, Dr. King, Michael King, Dr. Philip Knopf, and Dr. Cara Bren. And so I really enjoyed working in this side. Dr. King was all computational. And it was all programming, and I did awful. I realized it was a great experience because I realized I am not a computer programmer, and it was a really bad experience. I got the technician, he did all of my assignments for me. I would just say, oh, this is what I need done. He would do all of it for me. He'd write the code, and I would sit there in front of the computer. I didn't get anything done. But the other 2 labs I had great experiences in, and I picked Cara Bren's lab, so she's all the way on this side, and she was an amazing mentor. I'm really lucky that I found her. So really, so what I will say is, I'm going to say it in a little bit, that when I picked my lab, a lot of people ask me, like, how do I decide which lab to pick? How do I decide who to do research with? And I always recommend picking based on the people who are in the lab. So I usually pick labs and experiences based purely on the people, and not at all on the research. And I think I've had really good luck with that. And the reason why is because if you're a person who's a scientist and you love research, whatever project that you end up working on, you're gonna kind of fall in love with it because it becomes your project, and you're gonna work on it every day, and you'll learn the techniques, and you'll get really good at it, and you learn to love what you do. The people you can't really change, right? So my makeup of who's in the lab and your PI, those are really, I think those are more important to pick a good fit and a good match, because you're gonna learn from these people, you're gonna work with these people every single day, you're gonna spend 10 hours a day with them, like you're gonna really wanna like the people. So personally, even more than the PI, the people in the lab was super important. So for me, it was a decision. Dr. Knopf had 2 older lab techs who had been there for like 30 years, and then 1 grad student. And so that's who I'd be working with all day. And Cara had 6 or 7 graduate students who were all really friendly, and just, I could tell I would be able to enjoy myself and learn a lot from them. And so that's the lab I picked. I was not interested in her research at all. I could barely understand it. I did end up falling in love with it. It was my project, right? And so that's what I would really recommend, is picking your lab and your jobs and your experiences based on the people, because that's really, really important. I wanted to include some pictures of me, so these are actual pictures of me that I had to dig up that were me in graduate school. That's me, the center one, that's me giving a presentation to the president of the American Chemical Society. I won the travel award, and I got to go to the American Chemical Society and meet the president of ACS. That was fun. That was the head of the Women's Chemists Committee. So that's me giving my presentations. So here's that slide I was telling you about. So finding the right lab, and it really is about the people. Make your project compatible. I'm not saying you can work on something you hate, right? There's some things that you might really not like. So you definitely want a project that you're gonna like in a field that you're interested in. But I think it really is, as long as it's like something you could see yourself doing, the people is so, so important. I also picked a really good PI. PIs are important. To me, you have to know your own personality and your own style and find out who you're gonna match well with, because there are hands-on advisors who are gonna work with you a lot, and there are hands-off advisors who won't work with you at all, and there are advisors who are famous, but they're gonna never be there, and there are advisors who are micromanagers and wanna know every single thing about everything you do. You need to know what's good for you. Some people love it when their advisor's next to them, doing experiments with them. I did not want that, but I did want an advisor who was really communicative. So I picked that perfect balance for me, which was an advisor who could really teach me, and she was very patient and was really she was just a born teacher, but she let me do She did not ever, I don't even think I saw her in the lab once, right? So she was not hands-on and not a micromanager, but she was a good teacher still. So to me, that was the perfect balance. And so you really need to you need to know who you are and what style you learn best with and find that balance. In grad school, I really knew it was important for me to find good balance. I joined a dance team, which for me was like just pure fun, right? I got to go dancing. We danced on the weekends, and I made good friends who I would have never known them. They were not my style of people or people who I would have ever met. And I became really good friends with them because just through this dance group. And it was so important for me to just like, I don't even think they knew anything about like what I was doing, that I was getting my PhD. It was pure, just like, let's just talk about social stuff and have fun. For me, that really helped me with the balance of graduate school. I took up running. I actually hate running still, but I do it because it's good for me. And I did. I took up running, and to me, I think it was important to be able to go running and just physically do something physical so that I could stay alive during graduate school. And then I had good friends. So that's Cara, that's my mentor. And then the other 3 women are the women who were in my research lab. We still see each other every year. I don't think we've gone like more than a year without seeing each other, and I'm still really good friends with them. I really seriously thought about what were some things that I would recommend to people. Start writing your dissertation early. So start writing it like right away, and not like the first day, but the earlier you can start, the better. Because to me, I wrote as I went along, and then it only took me about a month to write my dissertation because I had written all the 6 out of the 7 chapters already. So I just had to write 1 more chapter. So to me, it's worth it to start writing in advance. It's just less stressful. Read and write and learn from your mistakes. So my professor, my advisor would say, she would rip apart my writing because I didn't know how to write scientifically. So she'd rip it apart like I kindly do to my current students. You rip it apart, right? And the reason why you do it, it's not to be mean, and it's not just to make it better. It's so that my students learn how to write. And so what I would do is I wouldn't just accept all the changes. I would look and see, okay, why did she pick this wording over this? And I really tried to learn from the editing. And she would tell me, she says, I always will remember this because I was upset about her editing. I was like, oh. It's just always red. It's like, you must think, it made me feel bad, right? Because it would be all red with corrections. And she would say, well, you do know that it would take me a fraction of the time to write this myself. I do this so that you could do it first and you learn to write, and then I edit it. She's like, in all the time that it takes me to edit your stuff, it would take me just way less time for me to write it from scratch. But I do the process so that you learn. So that was really, it made me really think about the process and appreciate it. Use a reference organizer like EndNote. That's just kind of a practical thing. Keep an annotated bibliography. I like to do this so all the journal articles you read, because just the main thing is because your grad school is going to be 6 years, 5 to 6 years, and you're not going to remember the journal articles you read your first year. So if you keep an annotated bibliography, when you write your thesis, you can just pull from that bibliography. So that's something I'd recommend. Take opportunities that will help you build a portfolio that you're passionate about. So do other extras. Do extra things, not because you have to, but because you want to, because you're excited about it, and because you know that it's going to help you be a better scientist or a better thinker or a better researcher or a better teacher. So take advantage of, I did a bunch of stuff that, some of the stuff really I probably could have done without. It was a little bit of a waste of time. But most of the things I really think I learned, every little thing I did, you learn from it. Keep your CV updated. I just think that's good advice for everybody. Network. So the people who went to grad school with me, they're winning all these awards, and I'm Facebook friends with them, and they're becoming famous scientists. Peers will eventually become your colleagues and will become maybe sometimes famous colleagues, so keep in touch with them, and it's always good to network. Keep a really good notebook. That's just important because to me, I kept it really, really organized notebook, and it helps. Don't put all your self-worth into your research. This is super important because you're gonna fail and do really bad in research and make mistakes, and if all your self-worth is in your research, you're gonna feel like a failure 90% of the time. So don't do that. Just put your self-worth into yourself and who you are as a person and your personal growth. You're learning in grad school. Even though you're failing, your experiments are failing, you're growing and you're learning from experiences. So your experiments aren't working always, but you're always on the upward. Can you go backwards? I don't think you can like stop learning or go negative, right? You're always gonna be going up, right? Learning more and more and more. So that's a good thing. So put your self-worth in your journey and your experiences and not in your research. And then take care of your physical and your mental health. So after I was at University of Rochester, I did 1 semester of teaching just to get a little bit more teaching experience, as I was an adjunct at Colgate, and I taught some physics labs, and then at SUNY Brockport I taught some chemistry labs. That was a good experience. It was my first teaching experience. It made me realize that teaching is really hard, and it's something that you have to it's just like anything else. You've got to learn how to do it and figure out how to be a good teacher. That was my postdoc at Cornell. Linda Nicholson was my advisor. She's an amazing person and an amazing mentor. So the woman who's next to me in that picture, Sarah, she was an RIT student, and so she was an RIT chemistry graduate. And so she gave me a lot of advice when I was applying to RIT positions. My postdoc was probably one of the worst, not the worst experience, but I was mentally, I think I was at the lowest point in my life. I was very depressed. I went through grad school, surprisingly, with very little mental health issues, because I lived at home and I felt really supported at home, like I did a lot of extras. My postdoc was all about work. I only did work. And I think I really suffered. So that was my first time where I realized how important mental health is in addition to physical health and everything else. And I got a counselor for the first time. I went to go get counseling, and that I think helped. And I think it was a lot of things at once. But I always tell students, I tell them this story because I think, I don't want you to look at me and think, oh, it's been pretty easy, and oh, she's done all these great things, and there hasn't been bumps in the road. Well, no, I have had significant struggles, and I think, and I don't want to say ashamed. I had to tell myself, I need to reach out for help, because if I don't, it's going to be a mess. So you have to kind of put everything aside and say, I need to take care of myself, and I need to do what's good for my mental health. And there's no shame in that at all. That's the right thing to do. Grad students have struggled with mental health. I didn't experience it until my postdoc, but it's a very common thing. Don't be afraid to ask for help, because it's more common that you're struggling with your mental health than it is uncommon. So that's just kind of my PSA. Happy things. So this is my dog, Mr. T. I adopted Mr. T during my postdoc, and I think this is kind of the straw that broke the camel's back, in that I had recently gotten married. My husband lived at home here in Rochester, and we adopted Mr. T. He couldn't be left alone. I think he was neglected as well, so he couldn't, like the second you left him alone, like he would go to the bathroom everywhere and run around, and he would like slam himself against the wall. It was crazy, right? So this is like not a normal dog, but we loved him so much, and I fell in love with him. So I'm like, I think I need to leave my postdoc so I could take care of my dog. So to be honest, it was more like my husband was like, I can be away from my husband, but my dog, I can't be away from my dog. So I decided to move home and be able to take care of my dog. So I did, I left. So I really did, I shortened my postdoc, and I got a job as a contractor at Ortho Clinical Diagnostics here in sunny Rochester. And it was the right decision. I don't know if that was the right decision professionally, but it was the right decision mentally and emotionally for me, and I'm really glad I did it. Plus I loved working at Ortho. It was fun. I was only a contractor, but it was an amazing experience, and it gave me a little bit of experience in industry, which I find useful because a lot of my students want to eventually go into jobs with industry, and I can say, even though my experience was limited, I can say I loved working in industry. It was amazing. I worked on cancer research, the only time in my whole science career that I worked on cancer research, and I worked with Judah Folkman, who I looked up. He was like super famous. Right? And I didn't know who he was. I had never heard, because I had never worked with cancer, and he was super famous. And so I have 2 papers with Judah Folkman from working in industry. And so that was so wonderful. In industry, you work in teams. In academics, you work in silos. So yes, we collaborate, but you're doing your own project, right? You're doing your own thing within your lab. In industry, it's a lot, it's teamwork, it's more teamwork, right? Which I really loved, because you're all working towards the same goal. Obviously, it's a business and not about academics. It's not about the pure joy of learning and expanding our knowledge. It's about making money. It's a business. And they don't sugarcoat that. They want to make money, and so there's going to be less freedom, because they're going to do the things that they want to do, that they want you to do to make money. So we have less money in academics, but we have a lot more freedom to do what we want to do. There's politics at both industry and in academics. So all my former research students, they'll complain about the politics that are in industry, and I'm like, they're the same. There's the same in academics. There's politics everywhere. It's not the most fun part of any job, but they're there. Our products here in academics are our students. Really, yes, we do research, but it's really, my products is my students and their success. Products in industry is more like real products, actual products that you can make money off of. There's a lot more rules in industry. You think we have rules here in college? There's way more rules in industry. I did not like that. There's more regulations. I wasn't a fan of that. You don't have to worry about funding. I remember they would say if I would need to buy stuff, I would just buy whatever I want. So yeah, we worry more about getting funding here in academia, but we worry less worry about the bottom line. So the bottom line is really about like efficiency and like, yeah. So they do still worry about making money in industry, but it's a different kind of concern than it is in academics. So in general, I think there's pluses, and I would never say that academics or industry are better or worse than each other. They're just a better fit for different people. And I loved working in industry too. And I love more working in academics, but that's just my personality. So this is my first real job here at RIT. It's funny because you're a student and then a postdoc. You're just a student for so long. You know, I was 29, I was almost 30 when I got my first job. So I've been here this is my 10th year. And I started off as an assistant professor in chemistry. And so really that's the end of my academic journey. My title was from physics to chemistry to biology and back. I'll tell you about the and back later. But really, what's cool, so I was at undergrad, I was in physics. In grad school, I worked in a chemistry lab. And then as soon as I got to RIT, I started really focusing on biology. So I collaborated with Dr. Pakikro, who's the director. He's a MD, a pediatrician, and he's also the director of the Rochester General Hospital Research Institute. So that's been just an amazing experience. He's not the easiest person to work with, but he is an amazing, amazing mentor. He's taught me a lot. He's tough. He was tough when I needed him to be, which is all especially during my pre-tenure time. Judith Hellman is in the middle. She's probably one of my most amazing role models. She is from UC San Francisco. She's a medical doctor as well, but does a lot of research. And she's just an amazing woman, and she's been a great collaborator. I'll talk to you a little bit about the stuff that I do with her. Anna Konovalova. She was at Princeton with my collaborator, and now she's a brand new assistant professor. So she's kind of like a new collaboration, but she's amazing. Here are I'm just going to briefly talk about my projects. So my current projects right now, I have 3 major projects. And Kara has worked on 2 of my 3 projects, right? So just not crystallines. So one of my current projects that Kara is working on is focused on ear infections. So ear infections, to me, the reason why we got interested in ear infections is because Michael Picicro, who's a pediatrician, cared about ear infections. And 75% of kids get an ear infection, at least one, before they're 3. So it's really, really common. What's also common is recurrent ear infections, so getting ear infection after ear infection. So a couple of my research students actually had recurrent ear infections, and that's how they went deaf, from those recurrent ear infections, which is obviously a very serious, one of the serious cases that you can have. It's a $5 billion a year industry, just ear infections, because of and that accounts for the amount of money it's spent on visits to the doctors and the hospitals, antibiotics, and time off of work, because kids will get ear infections, and so they count that. So it's a $5 billion a year industry. And a lot of antibiotics are prescribed for ear infections. And nowadays, with our concern about antibiotic-resistant bacteria, we want to fix this, because it's a huge problem. It's the most common ailment for which antibiotics are prescribed. So if we can at least lower the incidence of ear infections, we can really help with the antibiotics. Our goal is to find bacterial proteins that we can use in vaccines to protect against getting ear infections. Ear infections are caused by three different bacteria, and I work on one of them. The problem is that proteins aren't super immunogenic, meaning that, okay, so let's say we have a beautiful protein from a bacteria that causes ear infections. We put it into a vaccine, and we give the kids a vaccine. They're not going to stir up the immune system that much, and so the proteins are they're so small. They almost are like the body looks at them and says, filters them and gets rid of them. They don't make that antibody response that we want for a vaccine that says, remember this, because this is a bad thing. And so those memory antibodies are the things we need to make with a vaccine. And so, our challenge, Kara's challenge, is how can we make our proteins more immunogenic? And so she's working on ways that we can amp up the immunogenicity it's almost like amping up the toxicity of these proteins, but the problem is that when we do that, they're harder to work with. So Kara works with proteins that are really hard to work with. It's super, super challenging, but it's why we are doing it, is because the payoff will really be really big, because we really need these vaccines. So that's my ear infection project. Yeah, PJ.
Is it possible to work with smaller peptide regions of these proteins that
Yeah, so we could, and we've looked at that too. We've tried to identify the most immunogenic regions within the protein to see if we could just use a peptide. But we have the same problem that even peptides are even smaller, and sometimes, even though they're still not super immunogenic. So, but that is one. I mean, there are scientists, I think, in Japan who are working on that, the peptides from the bacterial proteins. The most immunogenic parts. So we have, that was actually one of my first projects, was identifying the most immunogenic regions in the proteins. So it's a struggle every day, but we work really hard. We have a mouse model that we can test the vaccines with. I learned how to work with mice during my sabbatical 2 years ago, It's not the most fun things to do. Kara's come with me. It's not, I mean, working with mice is very challenging and hard, but it's important because the FDA will not approve a vaccine or anything like that unless it's been tested in an animal model, because they need to know that it's safe in living systems. So, it's a necessary, I don't want to say evil, but it's a necessary thing in order to move forward with our vaccine. So I knew it was an important part of my project. So I also work with sepsis, which is a full body infection. And this was Kara's first project, right? So this is a really exciting project that I work with Dr. Hellman at UC San Francisco. And sepsis is a huge problem. So I think that a lot of people, there's people who don't know about sepsis. Most people don't know about sepsis unless they've had a relative or someone who's gotten it. And they're trying to right now raise awareness about more awareness about sepsis. There's a big billboard, I think, on 390 that talks about sepsis and like awareness. So yeah, they're trying to increase awareness of sepsis. But really what it is it's full body inflammation. You get an infection, maybe you get a cut and you get a bad infection, and then the bacteria replicate and maybe they get into your bloodstream, or somehow they get, like there's a lot of them, and your whole body responds to that infection in a out of control way. And so you get out of control inflammation that can't be pushed back down. So that's what sepsis is this like whole body inflammation. And the problem is there's no real efficient way to diagnose sepsis, and there's no real treatment for sepsis. And so it's deadly. The question that Kara worked on is 4. Can we use PAL as a urine biomarker for diagnosis? We submitted a patent application, so it's patent pending. We haven't submitted the full application yet, but we want to use PAL protein, which is a bacterial protein found in one of the bacteria. It's called E. coli that causes infections, and we want to use it as a biomarker for early diagnosis of sepsis. So there's no way to definitively say someone has sepsis or even someone has this E. coli infection. So we want to be able to take someone's urine, which is fairly clean, there shouldn't be a lot of other stuff in there, and if we can find PAL in it, we should be able to say, oh, okay, this person definitely has an E. coli infection, this person has sepsis. So we're working hard on that, and we did a small pilot study on patient urine. So we got samples of urine from patients at RGH who had sepsis, and we were able to look and see, and we find the protein, and it would, beautiful detection of that protein in the urine samples. But it was a very small, limited study, so we're now applying very hard for grants to expand and make a large study. And then the last project that I work on is cataracts and crystallines. So I don't know if you've seen cataracts, but cataracts are basically just cloudiness in the eye lens. And so a lot of animals get this early on. Dogs are especially prone to getting cataracts. And really what it is it's just proteins called crystallines that aggregate and clump together, and then they prevent the light from being going through the eye lens in the appropriate way. And so one of the questions that we're asking is, how can we use our biophysics to understand how these proteins interact with each other, and why are they clumping up together? Maybe there's genetic mutations that cause more clumping, or the environment in the eye lens causes more clumping, or something like that. So we're working with George Thurston on that. The reason why I like working on it with George is a physics professor here at RIT, and it brings me back to my physics days, because he's a physicist and an amazingly genius. He's a genius. So it helps me remember my physics roots, and how I get to go back and think about physics questions. Other things that are important to me, my family, my personal life are also still important to me. My job is very, very important to me, but so is my family. So I have 2 girls. My 2 daughters are Charlie and Laurelyn, who are 8 and 5. That's my husband, Scott. So we went to Niagara Falls like a couple of weeks ago. That was fun. And that was from my cousin's wedding. So I think that it's really important for me to balance. I don't always do a great job of it, but I'm still working on it, balancing my personal and my family life with my work and research and students' lives. That's the end of my talk. Here are the people who actually do all the work, all the research. I was barely in the lab. I could barely do gels anymore. So the people who actually did all those experiments were my students. Kara was not here for the picture. Where were you? You were here. She was here for one of these seminars, so she couldn't be in the photo. But she's been in, actually I found, she's been in like 1 million of my photos. So, but here is my group, minus Kara, and I think maybe one other person. That's my research group. I have a big research group. I've had like over 50 research students, so I've had a lot since I've been here. So, all right, so these are my final thoughts that I'll leave you with. Everyone has their own journey to take. This was mine, but take your own journey. Don't let anyone tell you that you don't belong. I think that's so important, especially for my deaf and hard of hearing students, because I know that I've had experience where some of my students have gone on to graduate school and they were told by multiple people, which to me boggles my mind, but they've been told that they just don't belong there. And who is it? No one can tell you that, right? You do belong, and so don't let anyone tell you that. Science is tough, so you need to be tougher. We all know that, research is hard, so you have to just keep at it. Don't be afraid to ask for help when you need it, because everybody struggles sometimes, every single person. They're probably lying if they say they didn't have any struggles in their lives. And then you can do this because you're an RIT tiger, and you're gonna leave here, and you're gonna just know that you're well prepared, because RIT prepares their students really well. Our students are successful in jobs, in grad school, in medical school, in whatever they wanna do. So you are well prepared. Don't think you're not. You can do it. That's it. Thank you.